All right, welcome to Module 5 of the course. Today we're going to be discussing energy and cells and how cells obtain that energy, and also talking about how molecules move across cellular membranes. They'll do these in a variety of different ways, and this module is going to take you through all of those types of transport mechanisms, and we'll talk again how cells get their energy from sugars. We'll talk about cellular respiration and glycolysis, etc. The first thing we're going to discuss in this video is passive transport and tonicity of solutions, and how that's related to the movement of molecules across cellular membranes. And so we finished in the last module discussing cells and their various components. And one of the big components of cells, whether they're eukaryotic cells or prokaryotic cells, is that they have this plasma membrane. And molecules can move across the plasma membrane in a variety of different ways. One of those is through passive transport. And so molecules will move randomly because they vibrate, and that allows them to move through the air, through liquid, and they do so based off of a couple of different properties. And one of those is the process of diffusion. And so diffusion is when you have molecules to spread out evenly into a space. And so think about diffusion as whenever you spray perfume or cologne or Febreze, you'll spray it in one location in the air, and then those molecules will be concentrated in that location. And over time, those molecules will spread out to evenly fill that available space wherever you have sprayed it. That is the process of diffusion, and molecules can use that process to move across the plasma membranes. And this is part of the passive transportation of those molecules across that cell membrane, and this is done without any energy. The passive transport of molecules, so the diffusion of molecules, does not need any energy for that to happen. And what drives the movement of these molecules is the movement of the molecules across the concentration gradient. And so the concentration gradient says that molecules will diffuse from high concentrations to low concentrations. And so this is kind of how the molecules end up filling this space. And so, for example, in this figure here, we have red molecules, and there's this membrane that's separating the left side from the right-hand side. And on the left-hand side, this is where all of the molecules are concentrated. So they have a high concentration on the left-hand side of the membrane. They have a low concentration on the right-hand side of the membrane. So the molecules will diffuse from the left-hand side to the right-hand side until there's an equilibrium, till that space is filled. And so the net diffusion is from left to right until that equilibrium is fixed. And so you can have multiple diffusion or multiple concentration gradients for different molecules. So each molecule will have its own concentration gradient. So for example, in this bottom figure, the purple molecules are on the left-hand side, the right molecules are on the right-hand side. So there's a higher concentration of purple molecules on the left, higher concentration of red molecules on the right. So the purple molecules will diffuse from left to right. The red molecules will diffuse from right to left based on their concentration gradients, and they will do so until that equilibrium is set, until those molecules have filled that space. And so this is a form of passive transport. This happens across cellular membranes with small molecules that are not charged. And so remember, those plasma membranes are made up of phospholipids, and phospholipids have a polar head and a non-polar tail, meaning that the tails don't like to come in contact with charged molecules. So as long as those molecules are small enough to fit between that membrane and they're not charged, then they will be able to passively diffuse across that plasma membrane. And so the substances that don't fit that qualification, so that don't cross the membrane spontaneously, will do so via the transport of proteins. So you can get ions to diffuse across the plasma membrane, but they need to do so through the process of facilitated diffusion. And basically, facilitated diffusion is the diffusion of molecules across a membrane via protein channels. So basically, you'll have proteins in the membrane that will serve basically like a pipe or a tunnel for these molecules to move across. And the reason that facilitated diffusion is a form of passive transport is because they're moving, the molecules are moving without energy being used. And so example of those different types of passive transport are shown in the molecule or the figure here. So you have a cellular membrane, you have a non-charged molecule, which are the purple molecules. They have a larger concentration on the outside of the cell versus the inside of the cell. So those molecules will diffuse across the plasma membrane. Okay. Now let's say we have larger molecules, or we have molecules that are charged. They will move across that membrane via facilitated diffusion. So there's a protein with a channel. It will allow that molecule to move through the membrane through diffusion. Higher concentration outside, lower concentration inside. But this molecule is not coming in contact with the plasma membrane itself. It's going through a protein, which again is serving as like a pipe or a tunnel or a channel that's allowing that molecule to move through without coming into contact with that non-polar space. Same thing here. You just have a protein that will change shape based on the substance or that molecule coming in contact with it. And so again, this is passive transport because it doesn't need energy in order to move these molecules across the plasma membrane. In the next video, we'll talk about active transport, which is shown here, in which energy is needed to move these molecules, and these molecules will be moved against their concentration gradient. And so we'll talk more about that in the next video. Another way that molecules can move across the plasma membrane is through osmosis. And osmosis is the movement of water across the plasma membrane, and not necessarily so solute molecules like ions, so like salt and sugar. Osmosis is really important in the terms of water balance. And so the diffusion of water across a membrane that only allows water to move is called osmosis, so a selectively permeable membrane. And water will move across that membrane relative to a solute concentration. And so a solute is the amount of substance that's dissolved in a solvent. And so a solvent is a liquid. So a solvent is a liquid substance. Solute is anything you dissolve in that solvent, and that makes a solution. Okay. And so if water is the liquid solvent, then we refer to it as an aqueous solution. So for example, you have water that dissolves sugar. Sugar would be the solute. So here we have an example of the process of osmosis. So we have a U-shaped beaker with a selectively permeable membrane, and that membrane only allows water to move across that membrane surface. And so you have sugar molecules in this example, which are the green molecules. That is our solute. So you have low sugar concentration on the left-hand side. You have high sugar concentration on the right-hand side. So water will move from low solute concentration to high solute concentration. So in this example, water will move from left to right across that membrane. This net movement of water is osmosis. And so over time, more water will move to the right-hand side, causing the water level to rise in this U-shaped beaker. And so the water will continue moving from left to right until the water concentration is equal with the solutes in that solution. So for example, this right-hand figure, there's now equal solute concentrations compared to water molecules. So it's in equilibrium. So whenever you have equal solute concentrations, that is called an isotonic solution. So when the solute on the left-hand side is equal to the solute concentration on the right-hand side, that solution is isotonic. Okay, so those solute concentrations are equal. So let's say it's 10% sugar and 10% sugar. Now, if you have two solutions where the solute concentrations are not equal, so let's say the right-hand side has 20% sugar and the left-hand side has 5% sugar, then the lower concentration is known as hypotonic, which is the left-hand side. It only has 5%. The higher concentration of solute is referred to as hypertonic. That is the 30% or 20% sugar concentration. And so you're comparing one solution to the other and how much solute it has. So the solution with a higher concentration is hypertonic. The solution with the lower solute concentration is hypotonic. And if those solutions are equal, then they're isotonic. Now understanding these terms is really important because they're important for the process of osmoregulation. So the control of water balance in cells. And so water will move from low to high solute concentrations. And so water moves from a hypotonic solution to a hypertonic solution in effort to make those two solutions isotonic where they're equal, like we see here in this figure. Now, this is important, like I said, because of the process of osmoregulation, controlling the water balance within cells, controlling the water or the cell volume. And so the survival of cells really depend on its ability to take up and lose water based off of the solute concentration of the solution that the cell is in. So if an animal is in a hypotonic solution, it needs to be able to lose water because it will be taking up water. So if it's in a solution that has less salts than what's in the cell, then the cell will take up water. If it's in a solution that has more salts than the cell will lose water. And so they have to be able, the cells have to be able to balance that in order to maintain a constant cell volume. And we see figures here that are showing those examples. And so for example, if you're an animal cell and you are in a hypotonic solution, so hypotonic solution meaning that there is less solute in the solution surrounding the cell, then that water will move inside the cell. And that cell will swell with water and it could potentially burst. The opposite is if that cell is in a hypertonic solution, then water will move out of the cell and that cell could shrivel. And this is the case with animal cells because they don't have cell walls. For plant cells, the plant cells do have cell walls, and a hypotonic solution will lead to a turgid cell, which is a normal plant cell. So the vacuole in that plant cell will swell and it will push on that cell wall, causing it to be rigid. However, if the plant is shriveled and wilted, then it will be in a hypertonic solution, and that plant cell will lose water and cause that cell to shrivel. In isotonic solutions, you'll have a normal animal cell, and you'll have what's called a limp or flaccid plant cell. So it's not wilted, but it's getting to that state. So plants are happy under turgid conditions, and animal cells are happy under these normal isotonic conditions. And so we've already mentioned here that plant cells have those rigid cell walls. They're healthy under hypotonic environment where the net inflow of water is moving into the plant cells. And so that cell wall prevents those plant cells from bursting, whereas animal cells will burst because they don't have a cell wall. So if we look at these questions here, pause the video, try to answer these questions, and let's see what you can come up with. All right, so with the first one, it says an animal cell shrivels when it is blank compared to its environment. So if it's hypotonic compared to its environment, meaning it has less solutes, then it will shrivel because the water will move to the hypertonic environment. The cells of a wilted plant are isotonic compared to the environment because they're limp, they're going flaccid. And so that's isotonic, where the water is net flow is equal in and out of the plant cell. So here you have a turgid plant where it's standing upright. Left hand side you have a wilted plant where it's not turgid because of net movement of water is not into the plant cells. Okay, and so this is the overall discussion of passive transport and tonicity. Tonicity, I should have mentioned, tonicity is the comparison of the solutions to one another. So each of these are a type of tonicity. So you can have a hypertonic tonicity, a hypotonic tonicity, and an isotonic tonicity. And it's important to know how those tonicities dictate the flow of water and how that affects the water balance in animal and plant cells. And so in the next video, we will talk about active transport and talk about how active transport is different from passive transport. So we will see you in that next video.